And without further ado, I would love to introduce our second speaker today, Ariel Jaimovich. He is the Associate Director of Bioinformatics at Guardant Health, and he will tell us about the clinical utility of a multimodal circulating tumor DNA assay for early detection of colorectal cancer. Ariel, as soon as you're ready, feel free to take over. Thank you for being here tonight.
Thank you for the invitation, Monica. I'm happy to be here. Let's make sure I share the right one. Can you see the slides? Yeah, all right. Yes. So I'll be, talking about very similar things to what Nathan was talking about, but from a slightly different perspective. And I want to start by saying I am speaking here, but I'm actually representing an awesome team of scientists working together and sometimes also hanging outside in the sun once that was allowed. So you can see the picture of many of them here. And I want to give special credit to Drew, who was my kind of the driving force on the wet lab in making the assay I'm going to talk about. Victoria, who is our friend in medical affairs, working on all the collaboration, getting us samples, working on kind of the clinical interpretation of the data. Yupang and Oscar are working on the computational biology side, building the methods that allow for interpretation of the data, and Will has been our scientific advisor for a long time now. So I'm here basically representing the work these guys are doing, and it's been really a pleasure working with everybody here for the last 4 years. So I think this is needless to say here, but I'll maybe start with this. What is liquid biopsy? So basically, liquid biopsy is looking for cell-free DNA, and here we're also separating cell-free DNA that is circulating versus just tumor cells. And Guardant has been in many ways the pioneer in basically looking at this type of signal. But I think the one thing that stands out about Guardant Health is that we try to focus on use cases where we think that there is two things that are happening. One is that technology is really mature and ready to bring basically a product that is clinically helpful for patients. And that the second part of it is that the physicians and the patients are ready to use it in the right way. And I think that's been a key to what Guardant has been doing. And though I think the end vision is shared between what Guardant, Grail, Freenome, you name the liquid biopsy company you want are doing, something that has been unique about Guardant is in some ways taking baby steps towards that end goal and building things as we go that we believe will improve patients' life. And while doing that, bringing data to Guardant that we can use to develop the next iteration of products. That's, I think, the kind of the motto of the company. And so I will start, before I go into the epigenomic side, to talk a little bit about how Guardant started. And we're basically looking at, I will go through this continuum around the talk, but I will start actually with the first thing the company did, because again, that was at the time where the company felt that the technology is ready to bring a change in patients' life. And that was around treatment selection for late-stage cancer. So the question there is, the patients have advanced late-stage cancer, and they want to know what is the best therapy they can get. And at the time, most of the way that was done was just either using individual tests for individual genes, and most of them were done on a tissue biopsy that is, as you know, invasive, and in many cases has many technical issues. And that happened as I think now it seems kind of trivial, but 10 years ago, nobody thought this was such a huge problem. Now, when someone has late stage cancer, the question of what type of therapy should he get? Is it just a general chemotherapy? Is it targeted therapy to the specific mutations in this tumor? Is it immunotherapy? That is now a non-trivial question, and one that can have a great impact on the response and the side effects this patient will have. And what Guardant did is basically delivered an assay that is minimally invasive. So you get poked a little bit, but you only need to give blood. And we show that it's as efficient as the more complicated tumor tissue genotyping assays. And I won't go over all the Guardant 360 data. I just want to mention one, I think, pivotal study we have, and that's called Nile. And that's basically, as the company began to build this liquid biopsy assay, early on, we also, based on the belief that we really believe that this assay works as well as we publicize, a clinical study was launched early on and reported at April 2019, basically when people were got head-to-head on the same 280 patients, they got both a standard of care tissue genotyping as well as a Guardant 360 liquid biopsy. And what that assay that we were blind to showed is that the Guardant Health liquid biopsy test is basically concordant with tissue biopsy. It's faster, so we can give results in 7 days, which is much faster than most tissue genotyping companies do. And it's comprehensive in the sense that we cover almost all mutations that can guide treatment in these late-stage cancer patients. So basically showing that this is a very useful tool in the oncologist toolbox. And also recently, we've been the first, Guardant 360 was the first FDA-approved liquid biopsy for comprehensive tumor mutation profiling across all solid tumor cancers. So I think that's kind of where Guardant started. And there's been a lot of lessons and a lot of data generating during this effort. And this is what kind of our company stands for, is we do this while really helping people get better treatment to where they are. So after that quick introduction, let's switch gears towards early stage disease. And the first product I'm going to talk about is actually funny timing, because this was actually launched as a LDT only last week officially. So we had a few clinical trials launched earlier last year, but the official launch as an LDT happened only a couple of weeks ago. And this is a product that me and my team built during the last 2 years. And basically here, the question is, this is early stage disease where these patients are diagnosed with, in this case, early stage colorectal cancer. They go through treatment, so that can be surgery, sometimes followed by additional chemotherapy. And then the question they ask is, should they get another treatment? And I think the main reason why we chose this, and I will touch upon this here because I think it's important, is when you start moving from late stage cancer to early stage detection, I think the clinical question on what is the use case of a positive call becomes very complicated. In the sense that you as someone who provides the service, you want to be confident that the way the results are interpreted and used are really ending up, improving the patient's life. And as you move towards early stage disease, that questions become more and more complicated. What is the best use case? Can you prove that a positive call is really something that the physician should act on, versus maybe there's something there, but you probably shouldn't touch it. So how do you know that answer is something we're focused on? And the interesting thing about early stage colorectal cancer is that that is a very unique clinical use case where that has been answered. That in the sense that there's many works suggesting that in this specific setting, after surgery of early stage colorectal cancer, the existence of residual disease is indicative of a later relapse. So if you really, and furthermore, giving some sort of treatment, usually chemotherapy at this stage, can really not only kind of postpone a relapse, but really cure the disease. So the advantage with this specific use case is that we are pretty as confident as we can be. Of course, there's never 100% certainty where we live, but we're pretty confident that really the implication of a positive call here has a clear result for the oncologist saying, oh, we did a surgery, we do a Guardant Reveal test. If it's positive, we should give chemotherapy to this patient. And we think that this will really improve the type of treatment they're getting. So this is the clinical use case. What's the technology behind it? So again, I will start with non-epigenomic and then move to epigenomic, sorry. Basically where Guardant started is by looking at mutations as indication of cancer in cell-free DNA. The problem as you move to early stage disease, where you use this to actually call the presence of the disease, then there's two issues with genomic mutations. One is that many works have shown that using just these mutations will limit your sensitivity at 50%, and at earlier stages even lower. And for example, like Nathan discussed in breast cancer, it's probably going to be worse. And the other is that many non-tumor sources, for example, CHIP, and I'll talk about that in the next slide, can really confuse your analysis. So I'll touch upon this and then I'll shift to epigenomic signatures. But this is where we started. So I'll describe in one word for those of you who don't know what is CHIP. So CHIP is short for clonal hematopoiesis of intermediate potential. And this is basically considered to be also a precursor of hematological cancers. And this happens in the bone marrow where specific mutations, you can see these illustrations from this paper, where specific mutations happen in specific clones of the bone marrow. And some of them just take on like significant clones in the blood cells. And when they are in this CHIP stage, what you'll see if you sequence the cell-free DNA is they will look like tumor mutations, because they also happens in many cases in the same genes, in TP53, for example, but others as well. In many cases where you think, so around this 2015 period, when people started calling tumor presence by these mutations, they discovered they have many false positives, because these CHIP mutations, if you look at healthy individuals that are 65, 70 years old, the probability of seeing a CHIP mutation is very high. So in many cases, this can drive a false positive call. And basically what we did is, this is basically the first use we had of the Guardant 360 samples. So we have by now over 150,000 patients of late stage cancer. And using this data, we generated a filter that says, using the information we can learn from the data we have in-house, can we characterize what a tumor mutation looks like that is not CHIP? And basically, now we get all the mutations from the cell-free DNA. We filter them using this tool we generated, this classifier, and then we only use variants that we believe with a high likelihood are coming from the tumor, and only use these to call presence of an early stage disease. And to test this hypothesis, we worked with collaborators in MD Anderson, and this is a clinical set of patients with CRC, patients that went through hepatectomy. So they had a met in the liver, and the liver was removed. And then they took a liquid biopsy after surgery and asked us, can you use this to identify the kind of relapse. So what you can see here is if you ignore everything I just said about CHIP, and you say any mutation you see in cell-free DNA is indicative of tumor presence, then out of 51 patients, you will find mutations in 43 of them. So out of 51 that, and then that gives you, sorry, I'm looking at the wrong row. Out of 36 patients that relapsed, you will find this in 31 of them, you will find this mutation. And then that's 86% sensitivity for actual relapse of the disease. However, if you look at the specificity, so how many of your positive calls are actually non-relapsing, your specificity is pretty poor. So 6 out of these 15 will not relapse, which you can see also on this curve. I'm sure many of you have seen before, where this is the time that has passed since the surgery, and this is what percentage of these are still recurrence-free. And the orange line shows you every time a patient relapses, it goes down. So the orange line has positive calls, and it does, you can clearly see a difference. So these are clearly top populations, and yet it's not a perfect tool. So what many researchers do is they use tumor mutations. So basically what you can do is, okay, if you have tumor tissue information, what happens if you only use a tumor positive call and use that filter as only reporting from plasma things you also see in tumor. So what happens is that your sensitivity drops significantly, right? So you can see two things here that are actually very typical. One is the denominator goes down, so from 36 to 31. And that's because for 5 patients, you don't even have tumor information. And the other is that it drops your sensitivity because in many cases, you called it based on a CHIP mutation. But it becomes a very useful clinical tool because the specificity is perfect, right? So all the patients you report will relapse. And that gives the oncologist confidence that it can use this to assign chemotherapy without damaging the patient. And what we did is basically develop this, but in a way that's tumor tissue agnostic. So we used the filter I described in the previous slide. And with that, so here the denominator is the same because we use the same, we don't need the tissue. The sensitivity is a little bit lower than here, but actually similar or a little better than tumor tissue. And most importantly, the specificity is perfect. So by using this filter, we get to a point where we think we make this tool a useful tool for oncologists. So this is how we solve the CHIP problem, but this is where the epigenomic side comes in. The sensitivity is still not good enough. So we want to get the sensitivity better to really make it not only useful, but really, a powerful tool that can help more patients. So Nathan already talked about this a little bit in his talk, and as he mentioned, also this is very powerful, especially for colorectal cancer. That has been shown that you have differential DNA methylation. It actually exists in 2 different ways. One is if you look at the tumor, there's a global hypomethylation across many places. But in addition, there are specific places, many times in promoters, genes that are silenced, and other specific genomic locations in which there is a specific hypermethylation of these promoters. So you can actually use, if you know where to look for, you can detect tumor DNA just by looking at this methylation signature. And basically, what's unique about the Guardant Reveal assay that I believe, to the best of my knowledge, doesn't exist in any other company, is that we developed an assay that can query in a single assay both genomic alteration, mutations, and also methylation without kind of impacting without having methylation impact genomic alterations and vice versa. So how do we do that? Basically, the assay, after extraction of DNA, we partition the DNA into methylated DNA and non-methylated DNA. We barcode each partition with a different barcode set. After that, we use a panel of regions we think are informative either on the methylation side or on the mutation side to do a target capture to the specific regions we care about. We sequence the library, and then using the barcodes, we do separate analysis for both the methylation as well as the genomic alteration. For the genomic alterations, as I said, we apply a variant filter to make sure we don't get any chip mutations. To the methylation, we do a signal processing analysis to identify regions that are differential methylated in the blood, and then we combine the two calls to issue a final tumor detected versus not detected output. So the first piece of data I want to show you from this assay is basically kind of our way of looking at the data and making sure that it makes sense. So what do you see here? You see data from different patients on the y-axis. So every row here is different patients. The top half shows you cancer patients. The bottom half shows you healthy donors. So some of them are colonoscopy screened. So you're confident they're negative. Some are self-reported, so they with lower confidence, but still probably healthy. And then every column is a region in our panel. So the first set of regions you see here are regions we added to the panel as a negative control, as a positive control, because we believe they are hypermethylated in kind of ubiquitously in the genome. And we can really see here in all the patients, we find, so the heat map basically shows you how many reads we found as methylated as a percentage from all the reads we see in this region. So in this case, 100% of the reads are methylated. And in these columns, where these are regions we designed to be hypomethylated ubiquitously, we also see that almost in all of them, we get 0% methylation. Where does the signal come from? So we start by looking at regions that, based on the literature, we know that they have differential methylation in cancer compared to blood. And what you can see here is that we really find differentially between the cancer samples and healthy donors, that you find these regions really to be methylated by our assay in many of the patients. And furthermore, by using, again, data from our kind of the Guardant 360 biobank, we were also able to, using whole genome sequencing, identify many other regions that we find to be differentially methylated between cancer patients and healthy donors. And by addition of these regions, we basically have a very large, kind of many shots on goals, many regions that we think any type of differential methylation in them will be indicative of cancer versus healthy donors. And one more piece of evidence to kind of convince you that this is coming from the same biological source is that in our computational output, in addition to a positive versus negative call, we also output an estimation of what is the epigenomic tumor fraction. So, for example, if there's a region in which you expect methylated DNA to come from the tumor, and you find the methylated DNA to come from 20% of the reads you find, then you think that the tumor fraction is 20%. On the genomic side, this interpretation is much simpler because you just use the mutation math. And what we find is that this epigenomic tumor fraction on the X is actually very well correlated with the genomic tumor fraction on Y. And there's a few things that I like to point here. These are, by the way, CRC here in this orange-brown color, Lang in blue, works in both. I will not talk about the blue ones. So focus on CRC for this talk. A few things that are interesting to see here. One is that when the tumor fraction is high, this is very well correlated and on the diagonal. However, when the MAF is low, you can see this cluster of points here. When the MAF starts becoming less than 0.1%, this is where you see most of the points where very few calls are done here on the genomic side. And there's a lot of power driven here by the methylation caller. So the conclusion here is one, yes, we are very confident that the methylation signal is coming from the tumor and that it is adding power to the classification. How does this work in practice? So this is a collaboration with MGH, with Ryan Corcoran. And here we take patients that, early stage patients with CRC, and then we get blood that is either taking after the surgery or after adjuvant therapy they got after surgery. And based on one single draw of blood, we use it to identify tumor presence. And here you can see same type of statistics I've shown you before. This is the recurrence-free survival on y-axis and days in y and in x. And the separation here is between, this is the patients in which we detected the MRD. These ones, we did not detect anything. So again, even after addition of methylation, our specificity is perfect. So all the patients that we called as ctDNA positive ended up relapsing. And the sensitivity is better than genomic only. So you can see here that there's, in this case, 3 patients that had were detected only based on their methylation signal. And so we are very proud that this is, as I said, launched a couple of weeks ago. And again, the emphasis for us is to build things that can be launched and used right now and help people get their treatment. And we really think this can be the case here. So with the time I have, I'm going to switch quickly to the last application. So this is pre-cancer. So can you go from kind of looking at the case where you know that disease existed to a screening scenario in which the question is, this is basically, do I have cancer, right? So in this case, the holy grail of, excuse my French here. So here, maybe I'll go quickly through this, but I don't think there's a need to, in this crowd, explain, but really there is a need for screening, especially in CRC. Right now, the most common tool is colonoscopy. However, many people don't do a colonoscopy because it's invasive and time consuming. And over around a third of the people that should be screened for CRC are not. So our goal here, again, the clinical use case is clear because the utility of the screening is proven, but the reality is that many people are not screened. So can we just help these people who don't want to have a colonoscopy get screened and detect the disease at an early stage where they can really be cured using the technology we developed. And one thing we add here is the fragmentomics analysis that actually Nathan went into length. So I will maybe skip the slide I had, but the difference between our Lunar Reveal and this product is here we basically combine 3 signals, again, with a single assay, genomic mutations, methylation, and fragmentomics. And I will not go over this since Nathan spoke to this. And I'll just go quickly over the results and wrap up. Here, to test this, we basically developed the assay, and we got a testing cohort of 105 patients with the diagnosis of colorectal cancer, and where the plasma was taken prior to surgical resection, and we compare them with cancer-free controls. We make sure the ages are correlated, and these are the same average risk population, and that the stages are comparable to what a screening population would look like. And I want to emphasize that when we do other tests here, we basically train our models. We integrate the calls into a continuous score of the probability we think the tumor is there. And then to blind ourselves to the testing set, we set the threshold based on a set of training healthy donors and a target specificity. So we decide on target specificity on a training set before we saw any of the cancer patients. And we target specificity based on how many false positive we allow on the training set. And then we use that threshold on our blinded test set. And when we do this, so this is a case where we target 95% specificity. Our test set specificity is very close to that, 94. And our sensitivity there is 88, where you can see here the stages, stage 1, 2, 3, and 4. And you can see here the difference between those 2 bars is genomic only versus genomic plus epigenomic calls. And you see that across the stages, not surprisingly, the contribution of the epigenomic signal is crucial to get to the numbers we need to get. Maybe I'll skip those. We can also look at different targeting specificities, and as you would expect, lower specificity would end up with higher sensitivity. I want to mention that since then, we actually published another set of a testing, so an independent test set of actually more. So in this case, we had about 113 cancer patients and 224 healthy donors. The sensitivity here was very, very similar. So we get 90% sensitivity at similar specificity. I want to, again, the epigenomic contribution is crucial. One thing I want to highlight here that is interesting clinically is in this case, we had the healthy individuals where most of them were unscreened, but we were able to get a few that were actually screened negatives. And it's interesting that your specificity, so this is the arrow seeker. If you look at the specificity, you actually get better specificity on the screened one when you compare them to the unscreened one, which may suggest that some of these false positives are actually people who have some sort of early stage disease, but they're just not aware of it. So this was interesting in our case. The last piece of data I want to show here is that if you look at our continuous score, where you show here the blue, every dot is patient. These are your screen negatives, your self-declared healthy, stage 1, 2 in orange, stage 3 in red. The reason I show this is to show that our performance is pretty robust to threshold selection. So when we target the specificity, you basically put a line on this y-axis. And the reason I'm showing you this is to show that you can actually go put the line on an actually very large range, and your sensitivity and specificity will be very, very robust. So we are pretty confident that the performance we get is not overfitted to a specific threshold selection. So to summarize, we developed a very unique and novel assay that enables the measurements of both genomic and epigenomic analytes in the same, from a single tube. As I said, the lunar reveal assay that was launched a couple of weeks ago can be used to identify patients with high relapse risk from a single post-treatment plasma sample. So this is not kind of multiple samples. This is one sample after surgery. Using that, we call the treatment. And finally, the lunar 2 assay we're working on can detect early stage CRC, and we are looking forward for its launch into the market to really help increase the screening for this disease, and again, help people get better treatment. I want to say in the end that we are hiring. So if any of this sounds interesting to you and you want to come work with us, either on the BI side, assay, actually across the board, shoot me a message or look at our webpage. We have many, many open positions, and we'll be happy to hear about it. So that's it.
Thank you, Ariel. That was great. Thank you so much for sharing